All right, so anyway, thank you for coming to our seminar. We have one more next week. Sean, you want to mention anything about that one?
I don't know a lot. It's about mammal functional morphology with lots of, I think, medical implications.
And that will be in person, right?
Yes, that person is coming.
Wonderful. Yeah. We'll probably have something similar with some type of snacks. We'll figure that out. The other thing is, well, hopefully everything's going well for you and study hard for your coming semester. Is almost getting close, right? So anyway, Angella Morehouse was one of my invitations. She's actually an alumni here. And you might tell us her years. What years were you here again?
1990 and 2000. That's a great year.
And Rick Anderson was her advisor. So Rick Anderson was the former chair and did a lot of invertebrate kind of things and stuff like that. But essentially, there's nobody else faculty-wise that are around it, is what I understand. And I really was impressed by some of your pictures and books, and I've always heard your name, and so I thought it'd be great to invite you out here. You work with the Illinois Nature Preserve, obviously, and maybe you could tell us a little bit about your career and your background and what you did at Western, and then
Sure.
give it a shot.
Sure.
Thank you.
I appreciate this opportunity. Thanks for spending your lunchtime with me. So I started here. I'm a local, so I grew up about and now basically the Kibbe Field Station. So I'm from Warsaw, and so a lot of my parents went here. I went here. I went to junior college for 2 years, and then transferred here. Initially, I transferred as a chemistry major because I thought that's where the jobs were, but my passion was always biology. And so when I started doing really poorly in my chemistry classes, I switched over to biology, and I just took a chance. It was rough. I got a degree. Basically, just my bachelor's degree in biology, went out there in the world, needed a job. I had like $30 in my bank account. So I ended up getting a chemistry job in a lab, worked on that for a couple of years in Keokuk, Iowa. And then I got flooded out in 1993. And my husband and I moved to Havana to teach. We had a teaching job there. And I just started volunteering with Fish and Wildlife Service, Natural History Survey, whatever I could do. And 5 years later, I finally, landed this job with the Illinois Nature Preserves Commission. I did an internship, kind of like a graduate student worker position with the DNR nursery. And that's how I got my foot in the door. But to actually get that job, I had to go back and start working on my master's degree. Well, if I was going to go back, I was going to take it seriously. They told me after I got the job that I could then drop out of master's school and not get my master's degree. But I said, why would I do that? I made the commitment. I'm going to do this for me. So for the next approximately 4 or 5 years, I worked full time and worked on my master's. So I was a transition, basically commuting from Havana when I had to. I did a lot of online and independent study courses to get my master's degree. My project was actually on the Illinois River. So I studied waterfowl and moist soil management at Chautauqua National Wildlife Refuge. So I was a wetland specialist. When I got hired by the Nature Preserve Commission, they somehow termed me a prairie specialist because I did a lot of volunteer work doing burning prairies. I had a pesticide applicator's license, so I was more terrestrial and not so much on the river. So they dubbed me a prairie person, and I became a plant ecologist for the state. One of very few. We have very few plant ecologists. I'm now trying to transition the last 10 years into an entomologist, so I'm more of an invert ecologist. We have no entomologists within the DNR, so I am serving that while not being recognized so much formally. I'm still a plant ecologist. I have to put that hat on every once in a while. So it's kind of different. We actually get to do a lot of flexibility. I actually do land protection. So conservation easements, protection of high quality natural areas is my main job. This is my side project. So this is kind of some of what I do for fun, for my hobby, and to be able to make presentations on some research, to get to know the sites that I work on that I protect a little bit better, because nobody else is doing anything to find out what insects are out there. So I wanted to do that. So Casey Carter is my, is the Natural History Survey. She's a pollinator ecologist, and she essentially is helping me out with this. I'm just not on the right.
There
we go. Okay. So Casey Carter basically does a lot of bumblebee ecology for the Natural History Survey. A lot of funding right now for pollinators is based on monarchs or bumblebees. With the rusty patch bumblebee getting a federal designation, there's a lot of interest in this. There's all these there's almost 600 bees in the state of Illinois, and bumblebees represent about 10 of those. So there's a lot of them being ignored. So I wanted to change that. So in 2018, basically keeping in some of our goals within Department of Natural Resources, I wanted to find out more about the pollinators that we have on our sites. So I picked 30 sites in West Central Illinois, and I basically visit them 4 times a year for about 60 minutes or so, using the path of least resistance. I mean, I don't go through brambles and everything, fighting my way. If there's a creek bed, if there's a trail, I take it. I do photography. Basically, my husband was a photographer, so I picked up a camera. It's quick and easy for me to take a picture. I also get to know what that plant is what plant that insect is visiting. So I have that record, and then I can put it on Bug Guide or iNaturalist to get help in identifying them. One of the things we want to do is not only find out what baseline, what's at our sites, but we also want to know if there's going to be an impact with our management, with our prescribed fires, with our brush clearing, with our pesticide applications. Are we impacting these insects? And so I set out to do a long-term study to basically replicate these every 5 years to see if I can find out if something's happening at these sites. I get a lot more familiar with these sites when I visit them 4 times a year. So here's my sites in Western Illinois. I actually live, like I said, near Kibbe, so that's Cedar Glen, CG. Mississippi River Sand Hills is another nature preserve right there for those of you who have been there. So in 2023, I actually transitioned over to voice recording. So instead of writing it all down by hand, I actually start using Google Voice Recording, and I talk to myself. So I basically wear a lanyard around my neck, and I talk to myself. This enables me to record a lot more data. So there's my old sheets. Basically, I was old school. I took out a little notepad. And unfortunately, every time I wanted to photograph something, I usually had to basically throw that notebook down, take the picture real quick, and move on. Because of that, I was not able to record every time an insect visited a specific flower. I'd only record all the different flowers that were visited by that insect. Now that I have voice recording, I record every single interaction. A lot more data, massive data set. Like I said, I carry a lanyard around me so I can talk right into it. I tried the side pocket, but the shuffle of my feet interfered with that, so that didn't work out so well. So for the purposes of this talk, I'm going to be talking about Heteroptera or true bugs. I wrote a book recently about true bugs, trying to get these recognized as being pollinators. They are not probably the most important pollinator, certainly not the most efficient, but they are visiting a lot of flowers, and I wanted something that hadn't been done before, so that was kind of my niche. So things about true bugs is they have this big long rostrum, which is a 4-jointed rostrum, basically a stabby thing that basically punches into the flower or to a plant or to another insect, and basically has 2 tubes, one that injects the paralyzing liquids and one that sucks up the fluid, kind of like spiders do. When they're not using that, they tuck it under their belly. So they can't retract it. They have to basically tuck it underneath their bodies. So this is kind of just a representation of those 2 years, the percentage of different taxa groups. So obviously bees are the most important, but when it comes to species diversity, flies and beetles make up about the same proportions by species wise. Now bees are a little complicated because it's very hard to identify a lot of the bees to species level. Some of them are certainly level to the genus level and sometimes the subgenus level. So bees probably represents more species, but there's a lot of them that are very, very similar. There's a little tiny sweat bees. They all look alike. That might represent 40 species. Might represent 20. I don't know. I do the best I can with my photographs and with what help I can get. But as far as the number of individuals, you look at bees, and bees represent 40%. So there are a lot of individual bees out there on the landscape. But bugs represented 9 and 8%. That is about the same as butterflies and moths. Now this is daytime, so obviously I'm not getting out at night. There's going to be moths visiting the flowers at night. But during the daytime, bugs are visiting these flowers at the same rate as the butterflies and moths. So just here's kind of, there's 13 different families of bugs that I actually did report on, 767 different interactions, 43 bug taxa species when I can get to, and 92 different flowers that they were visiting. So obviously there are some groups that are way more likely to visit flowers. That includes the assassin bugs, which was kind of unusual, ebony bugs, plant bugs, and stink bugs. So I'm just going to kind of use common names, since most of you aren't going to be familiar with the scientific names. But so I gave all this data to Casey Carter because she has the ability to do the statistical analysis. Like I said, I'm a field biologist. They pay me to protect land. They pay me to manage land. They do not pay me to identify bees in a lab. They do not pay me to run statistical analysis. So I've got this beautiful, huge data set, and people from Natural History Survey, Casey's not the only one. There's 2 others from the survey. They're actually making use of my big, huge data set to actually get publications out there. If they do a statistical analysis and write the publications and I just proofread them and add my information to that, it's a win-win for me. So here is one of the bumblebee networks that Casey had worked on. Just kind of showing you an overview of what this network analysis looks like. So you essentially have plants on one side and the insects on the other with these lines showing the connections between the 2. So here is the bugs. So here is all the different, 92 different plants and 43 different bugs and their connections. And just a close up, obviously, the thicker the line. So those links basically represent how many. So how many are going to there. So you can see which ones are associated, but you also see is there a preference. So on there, you can see that Original strigosus daisy fleabane is the most popular plant, which is amazing because it's a little tiny little thing. But the bugs actually love daisy fleabane. It's kind of a weed. We never plant it, it just pops up. And then these little ebony bugs are the most abundant insect that we did see. And there's a close association between them and this daisy fleabane, along with, for some reason, they really loved basswood. So Tilia was another one. I just came upon a low branch of basswood, and it was just covered with ebony bugs. And that's what was represented by that. That was one actual observation. So connectedness, so the proportions of the all the possible different interactions of the bugs versus the flower and how they kind of relate to each other. So some insects obviously are visiting the same groups of flowers. So a group of insects is visiting the same types of flowers, and vice versa. The plants are being visited by the same groups of insects. So basically, this is true for bees too. A lot of times there's very low connectedness because there's a lot of opportunities out there. When you look at all the possible opportunities, they're visiting a lot of different things. So if it has good pollen and nectar, they're out there visiting them. So these basically just some of the bugs, the lupin bug, spined assassin, and the little minute pirate bugs occasionally get on your arms and they will bite, but they're not very. They're just kind of a nuisance. And then those are the most popular flowers that seem to interact in those groups. So this is a module hub that she basically came up with, just kind of showing some of the Passionica sativa, that's wild parsnip, daisy fleabane, and slender mountain mint are basically the plants that were visited by the same hub of insects, whereas the other mountain mint, hairy mountain mint, wild bergamot, wild quinine, and Indian hemp were visited by different suites of bugs outside of their modules. So just looking at the bugs themselves, so the native brown stink bug, Eustachius, and Lygus is the tarnished plant bug. They had the closest relationship within their own module. They're also very, very abundant. And then the other 3 that I showed you before, the lupine bug, the minute pirate bug, and the spined assassin bug, basically were connectors. They seem to connect different flowers. So nestedness, indicator of the network in which a species with many interactions interacts with species with few interactions. So like while you're visiting daisy fleabane, you might be the only insect that also visits wild geranium or something like that. But weighted nestedness was moderately high, indicating a system where the plants used by specialist bugs are also used by generalists. So there's a lot of overlap. So if you take some of these bugs out of the system, there will be another species that will actually visit that plant. I'm not saying the bugs are going to be the primary pollinator for most any of the plants. I don't think the importance of bugs is in their pollination service, but there is some relationships that are very strong with some of these bugs. And I think there's a lot we just don't know about them. So these are 3 of the specialist bugs whose host plant was kind of missing. I didn't see a lot of Biennial Gara, the Mirabilis, this is the wild forklock, or rattlesnake master. And those plants are critical for these specialist bugs. So their numbers are going to be a lot lower because I didn't have those plants. So robustness, the fraction of the species that must be removed to cause a fracture. There's not going to be a lot of fracture because, like I said, the bugs are actually overlapping in the flower visitations. That wild parsnip is so important. I don't know why. I think the carrot family itself is important. Wild parsnip has a basically nectar source that's easy to get to. It's in the environment, unfortunately. I'm not recommending anybody go out and plant wild parsnip, but there are members of the same family that can be planted to attract those insects. Bugs represent a small proportion of those total. So what we're going to do in the future is to look at all of the insects visiting the flowers in the network. It's a huge, huge network, and Casey's computer could not handle it within 24 hours. So she's going to have to go to the main office in Champaign and use a larger computer to figure that out. So one thing about bugs is they all have different strategies of feeding, and they almost all have multiple strategies of feeding. So the assassin bugs, as their name would suggest, are predatory. However, most of those assassin bugs will also feed on pollen and nectar. And that was something new that a lot of people talk about. But my photographs show this. A lot of the plant bugs are known for basically jabbing into the sides of a plant stem or leaf and sucking the juices of the plant out. They will also feed on carrion, or occasionally are predatory. So there's a lot of different feeding methods of these bugs. Like I said, most of them are doing more than one. Scat, I mean, there's little holes. They're basically, that's a plant bug. You see those little brown spots there? That's created by sucking out the juices. This is an assassin bug down here at the bottom. That's clearly feeding on nectar. We always call those flower dragons, the jagged ambush bugs. Essentially, I've seen a lot of times when they will feed on nectar. Why they do this, I don't know, but I do not think it's a starvation situation. Okay, these insects, certainly in my yard on these nature preserves, have a lot of opportunity. There's a lot of different sources of food. I don't think they're choosing to feed on nectar and pollen out of starvation. So they have other opportunities. So why they're doing it, I don't know yet. Interaction evenness. Analysis of how equal the number of interactions are. So there are a lot of bugs out there. There are a lot of flowers out there. Basically, it was moderately high, indicating a lot of different bugs are visiting a lot of different flowers. Network specialization. Again, this is really tricky because some of the plants were missing. And like I said, some of the bugs have different forms of feeding. So within the whole entire bipartite network, specialization was considered moderate across the entire system, with neither real specialists or real generalists. Again, that's a problem. This is showing some of the nymph specialists, but their adults will likely visit other plants. In fact, a lot of times the adults never visit the host plant that the nymphs absolutely have to have. So here's just a list of some of the floral specialists. I basically assigned kind of my own common name for some of these just to basically represent which flower that they do visit. So a lot of these, almost all of these, have to have that host plant in order to rear their young. So they'll lay their eggs in the flower heads. Their babies and their nymphs will hang around those flowers until they develop. Then when they're adults, they may still, like in the case of the milkweed bugs, they will still hang around the milkweed plants, but the adults will also feed on other things. So there's just some pictures of the adults on top and then the specialized nymphs at the bottom. So the large milkweed bug, you see those really commonly, just even recently on milkweed seed pods. The adults will feed on other flowers, but you always see the little nymphs on the milkweed seed pods. Biennial same thing. Your stilt bugs, you see the adults really closely aligned with biennial gara, and the nymphs are always on the little flower heads. They're like little tiny green things. They blend in. You have to really know what you're looking for. The false milkweed bug, because it looks very similar to the small milkweed bug and the large, has to have oxeye false sunflower, Heliopsis, to rear their babies. And so if I see those bugs, I know that there must be Heliopsis, false sunflower in the landscape. It's a popular restoration plant. People put it in their prairies. And because of that, this bug is increasing. So the degree, the sum of all the different interactions. So this ebony bug was by far the most common. So it visited 27 different plant species. And daisy fleabane, again, the top plant, was visited by 35% of the bugs. So those associations were pretty strong. Effective partners. So involves analysis of how equal the number of interactions are in relation to the number of partner species. This indicates a species functional redundancy. So like I said, there's a lot of redundancy built in. I don't see any of these being a keystone species. So if we lose one, we're going to lose the whole system. The exception might be those milkweed bugs. There is a lot going on with the milkweed bugs, the 2 species of milkweed bugs that actually feed a little bit on the milkweed plants and a little bit the seeds themselves. I think that relationship is pretty important, and I don't know how it would impact milkweeds, but there may be some association there. So just kind of some of the important partners, the jagged ambush bug, the Plagionaphis plant bug, the ebony bug again, broad-headed bugs. Wild parsnip again is important, along with common and world milkweed. They were visited by the greatest number of bugs at equal rates. So pollinator service index. This is one of the things that we really, once we started doing this, began to realize that bugs are not giving us the whole picture, because there's all these other, there's the other, 91, 92% of all the insects visiting the flowers that we're not looking at here. So like I said, if you take these out of the system, it's not necessarily going to collapse. So the pollinator service index measures the significance of the pollinator species. The significance of these to the plants is probably pretty minor, but they are out there, they are transferring pollen, and they're actually really fun to look at. This is the biggest thing. The reason why I'm showing you all the pictures of these bugs to show off my photographs and to just try to encourage you to look at these plants, because the more I look, the more I find these things. These things are everywhere now. And I'm actually, my percentage is probably going to keep going up because I'm spending so much time looking for bugs. I'm learning how to look for bugs and I'm finding them. I would guess that a person just starting out trying to replicate my studies would not see as many bugs because they're not trained to do it. I've trained myself to look specifically for these bugs and I'm finding them. So these are the most abundant bugs out there. And those are the top plants. So homewort is another woodland plant. It had little tiny flowers, or really nobody really notices them because they're little tiny white flowers. They basically bloom just after the spring beauties and all the big important spring woodland wildflowers, but they are visited by a lot of bugs. Wild quinines included in a lot of restorations and plantings. You can find it in a lot of remnants. Daisy fleabane again is a weed that just kind of pops up along the sides of paths. And then your wild parsnip is an invasive species, but it's a member of the carrot family. So we wanted to try to find out why we were seeing the results we were. So we looked at a lot of different factors. So we looked at overwintering stages. The ones that are overwintering as adults are going to basically be available to feed on nectar and pollen earlier in the year than the ones that have to basically hatch out of their egg or develop as a nymph. We looked at the date and time periods they're active, because some of these bugs are only active for a couple months in the summer. Some of them are active from March through November. So that's a big, huge difference. But we didn't find a lot of relationship in any of those. Prolonged feeding period didn't seem to matter because they're feeding as both nymphs and adults. So the period of feeding is pretty long. What type of community they're found in? Again, forest, grassland, wetlands. Those communities tend to integrate because as we start clearing some of the brush, we still have forest plants that are hanging on. You basically just increase the sunlight for the homewort and the homewort continues to bloom. And so we have that flower available. We can't necessarily say that that's a forest community. It was a prairie community that got invaded by trees. So there's just a lot of integrated and overlap. But the 3 that we do think, and this needs to be investigated more, floral scent does seem to matter. Bugs basically have a lot to do with scents. Obviously, stink bugs are producing a scent for different purposes. So we think that the mints, carrots, and milkweeds actually have a fragrance to them. They're probably attracting the bugs. Flower color, most definitely. White and yellow flowers are preferred, and flower structure. So they're visiting the flat surface flowers or little cups, so they're easy to get to. So pollinator mouthparts. Obviously, some bees and of course butterflies and moths have very long tongues of proboscis that can get deep into these complicated flowers. Bugs have that pokey little syringe-like thing, the rostrum. So they just basically can stab at it. They don't get inside complex flowers most of the time. So again, the shallow depth of flowers. So you have your white and your yellow flowers. They're very shallow. Those are the ones they prefer, along with the small cup-like flowers. And very, very few of them will actually visit more complex, deep tube flowers. And by complex, I just mean that it's kind of hard for the pollinator to get in there. The flower structure is just a little bit more complicated. These big bell-like shaped things, obedient plant, they basically have to crawl down inside there. But those particular bugs, the twice-stabbed stink bugs, really like obedient plant and other flowers that are shaped like that. They live inside that flower a lot. They mate inside there. They do all these things. Turtlehead's another one. Foxglove, beardstongue, these big tube likes. The irises, you have to crawl down that tube to actually get to the resources. So the tarnished plant bug, the twice-stabbed stink bug, the clouded plant bug, and the tarnished plant bug are the only four that I have observed visiting these complex flower types. So future plans. In the future, I basically want to look at my entire data set, which is going to be a lot bigger than this 767 observations. So it's absolutely huge. So 9,600 different observations in the last 2 years. It's a lot of data. We're talking 566 different insects. So that basically that bipartite network is going to be absolutely huge, and we'll have to take it apart in pieces. 200 different plant species. It's going to require a much bigger computer, a mainframe to process all that. I'm going to continue to run these. I'm doing it on a 5-year rotational basis. So I visited of my 6 sites per year, 30 different sites, every 5 years. So right now I'm into just finished year 7. So this next year will be year 8. So I'm hoping to get in 15 years. So 3 cycles of 5 before I retire. We'll see if I go for 20 years. I don't know. Right now I'm thinking no, but assessing the impacts of land management. So one of the things we're looking at is sites that are intensely managed with prescribed fire, brush clearing. We're using a lot of forestry mowers to mulch up the excessive trees to basically bring the prairie back. We want to know the impact of that. So far, both of those exercises are actually pretty favorable because you're creating more sunlight. Anytime you get more sunlight, the fire stimulates flowers to bloom. You're going to get more insects as long as there's refugia around. So they're going to come back and visit on those flowers. We're going to continue to collect data. I have a lot of my photographs are all on iNaturalist and Bug Guide for the most part. And so I still get identification corrections and updates to try to get things down to species. As the AI on iNaturalist gets better and better, it's able to, along with experts' help, able to help me identify the species that I photographed. And so that's really exciting. If it wouldn't have been for iNaturalist and Bug Guide, I don't think I could have got into this because there's not a lot of good. I'm old school. I'd love to have a book that tells me how to identify things, but there's not good keys out there. Some people use Discover Life for the bees. That's really complicated, and there's a lot of bees out there, and I do not dissect them. So we need keys that basically help you identify it in the field. And so with that, this is my bug. It's been out for a year now. It's available from Pollinator Press as basically a field guide with a lot of photographs, and it talks a lot about the different plant associations. So that's just one of the things I've done. I'm kind of hoping to do flies next, but that's going to be a bigger book. So, and with that, I'm happy to answer any questions. Or I can talk more. Have any questions?
Okay, so how can you like tell how much bug is acting as a pollinator? You can see it visiting the flowers, and I guess you can see pollen on it.
Right.
And so do you have a sense of how well they're really transferring pollen?
Well, you'd have to wash them. You'd have to collect them and wash them, and get their pollen loads. And I'm not paid to do that, but that's the way you would do it. And that way you'd also find out what that pollen is. I mean, there's a lot of research out there to actually identify the actual specific pollen grains. So you can actually definitively say. They're also now doing, which is kind of scary, but they're doing pollen analysis to identify which bees are visiting the flowers based on the genetic markers left on them, as opposed to doing hardcore taxonomy. And so, of course, the bee experts are really nervous about this and don't like it. This is the Fish and Wildlife Service, and the federal government is investing a lot of money in that research. And they're thinking that that's a future, because then they don't have to look for bees. They just have to go out and collect the flowers and get the genetic marker on those flowers. And so it kind of does away with the hardcore taxonomy. And there's a big fear about that. Right now, it is not economically viable to do that, but because it's new and sexy, that's the way to go. That's where the funding's going.
What's the fear about?
Well, are you losing hardcore taxonomy? Are you using taxonomists? Nobody wants to pay the $2 per bee to have it identified in the lab. So it costs about $2 per specimen. And that's a cheap, just a round out figure. So there are basically in the Midwest, we just had meetings recently for bees. There are about 4 or 5 people that would do this on contract. They're really, really good. But there's no money to pay them. They don't want to pay these people. So these people aren't going to have jobs doing that. They're going to have to do something else. And losing taxonomy is a big fear of all of us field biologists. While I do photographs, I do appreciate the people that do the hardcore taxonomy. They actually collect individuals, vouch for them, and keep them in so that, you know, we have that to know, voucher to go off of, so we can help learn. We have that to go back and double check our work. We need that, and we lose our taxonomists. That's scary. I mean, I'm all for AI because it helps me with my photographs. I love the fact that AI can actually, the computer can actually pick out all the different features that we may not see with the naked eye. That's wonderful. That's exciting. But there are limitations to how far we should take some of these new technologies. Any other questions?
You said going into it that you are like going back and forth to online sites and different books to learn to identify them while you're doing the research. Is that like, I'm trying to figure out how to word this without sounding rude. Is that like a viable thing that you can do? Because
I'm getting better. I am getting better. And yes, yes, my data, that's what I can. That's the reality of it. I mean, it is what it is. Obviously, the more I do this, I mean, I was doing it for probably about 5 years before I started this project. So I had a little bit of background. I'm just not doing it from scratch. I was really good at butterflies. I've been doing butterfly surveys for about 25 years now. So I don't have to photograph butterflies. If I see a butterfly fly by, I just write it down or tell myself I have a, you know, what species I have. I don't chase butterflies unless I can photograph on a flower easily. But yes, I'm getting better. Is it possibly biasing my data? Yes, but it's what I have. So you can only get the data you can get. And that's just one of those conditions. But I think everybody's, all studies are that way. You're all going to get, always going to get better as you do it, the more you do it. And, you know, we can't wait around. Problem with the data is we can't wait around until we all become experts, because that's never going to happen. So you've got
Can I follow that? So I think I missed the first part of Kate's question, but if you're sending stuff to iNaturalist, I don't have it, but isn't there a community online that helps? There are professionals that are responding to the photos.
yes.
So, yeah, it's not like.
Oh, it's not just
me.
AI. Yeah,
It's
yeah.
like a professional bug guy that's
Yeah.
looking at his photos.
Yeah, I love it. The only bad thing about bug guys, it doesn't map. So you can't see specifically. So I use iNaturalist and I can go back to the map and I can see what I've seen there and what other people have seen there. It's really good. And then the ability to message people directly on iNaturalist through their message system. So you can actually get a hold of the experts directly, and they almost always respond. And the hardcore people that are on there every day, like myself, you know, although I don't do as many identifications, but I do respond to the messages I get. That's just a really cool tool. So I've met a lot of really good people. But for bees, there's some really good experts. There's still limitations with bees, but there's a guy in Singapore that he spends hours every day. He used to work in New York, so he's familiar with North American bees. But he spends hours every day identifying bees on both Bug Guide and iNaturalist. So if you posted a bee and John Asher has identified it for you.
So I don't know I've never seen any of this network analysis stuff, so I don't know any of those words. Could you show us your was there one figure that is your result, your plants and your bugs? Can you show us that?
That one, it's hard to read, and of course that network. That's the problem. They get so big when you got 92 species of plants, but that's the thing. I've got so many spreadsheets and they're absolutely huge. So depicting all that gets to be problematic.
That's okay. What I'm going to ask you to do is just, yeah. So I have a bunch of genetics students who have to write something.
Yeah.
So what I see here is you said, so the bug names are on top, correct? Okay, and the plant names are on the bottom.
Yes.
And then, so from this, you do some sort of analysis that gives you estimates of a lot of different things.
Right.
So can you tell me like two of the most important things? Like you started with connectedness, and then there was, can you just like remind me of like a couple of those things and where it is in this picture?
Okay, well, some of the things that are interesting, if I can, this is Lygaeus tursicus. This is the false milkweed bug. It's relative, Lygaeus calmi, that's a short milkweed bug. Those are the same genus.
Okay.
They're very, very similar. But look at where they are on there. They are not visiting the same plants at all. This one is only visiting a few, whereas this one down here is visiting quite a few, lots of rate. So that one might be visiting 15 plants, where this one's visiting 4. So that was something that was interesting.
Okay, so,
Yeah.
so I see it, so the size of the box is the number of photos you have of that taxon, is that right?
Now, there were times I recorded it, but I didn't always get a photograph, so I
Okay.
just told myself I saw it.
So that bug on the far left only has lines going to like 1, 2, 3, or 4 plants.
Mhm.
And its relative has a whole bunch of, so.
Yeah.
So from this, is there a name for that? Is that specialization?
Yes, this
That
one,
one-
this is the one that absolutely both, actually, they're both specialists. Their nymphs both have to have a single genus.
Okay,
So Lygaeus, it's Heliopsis, which is one species, whereas the Lygaeus calami, the small milkweed bugs, can use any milkweed. They prefer common milkweed, which is the most abundant.
okay,
but
so I
they
think,
have
so
to have
I'm trying to help me in this, so. One thing you can get from this picture is an estimate of bug specialization by seeing how many lines are coming out.
Right,
of it.
that whole statistic stuff that she generates, which, is done in the computer. The other thing that kind of surprised me is how thick some of those lines are. So even though Plagionasus visits, 25 different plants, it really, really liked homework. So that was something.
So yeah, so you were identifying for the first time the preferences of some of these
Yeah,
bugs.
yeah.
And how general or specific they are, right?
That's what I see, because I don't have the computer brain to do all the statistics in my head. This is visually what I see. I was really surprised that some of them are so separated from each other based on the plants that they visit. So
Right.
and obviously the ones that are only visiting, a few things, well, there's only a few of them seen.
So what a lot of the stuff I've seen, if there's some bug species that are frequently on flowers and other ones that you don't see very often.
Now, I removed all the ones that were singulars. So all the ones where there was only one bug visiting one flower were removed. So these all had to have at least 2 or 3 bugs that were seen by me.
Bottom one is the big black boxes where some plants receive more bug
Right.
visits.
Right. So yeah, there's this daisy fleabane and the homewarts and the wild parsnip.
Okay, so now, thank you. Can I ask another one? Yeah. So now I was gonna ask about the data. So for me, that's the kind of stuff I think about too, like preference. Do they
Right.
prefer things? Because, I mostly do parasitology stuff and it's like, what host do they use? So, your data, so I'm thinking about preference. Do you have data on like, you go to a field, plants are on this one, but, bugs are on this plant, but not on this plant? Do you have data for where bugs do not occur?
I do
That's
not collect null data because I just don't know what to do with that.
Okay.
Are there flowers on the, I originally started out by writing down all the flowers at the site.
Yeah,
And some of those site flowers are not visited, but
Okay.
because of the massive data set, 9,600 in 2 years, I do not have null data.
okay.
Essentially, it's just not practical to collect that.
And that is something that people study, right?
Sure.
Or they are on these more often than they occur.
Mhm.
So you have photos, your data are, if a bug's on a plant, what's it gonna be on?
Yes,
That's,
right. Now, I saw more bugs out there that I recorded, and some bugs certainly were on the plant previously, but if I didn't see it on that plant, it didn't get recorded. So that's especially true with some of the butterfly species, and I record all butterfly species, but so just for example, a question mark butterfly actually prefers scat than flowers a lot of times. So even though I record that question mark, scat is not a flower, so it's not going to show up on this thing. So there's certain species of butterflies that very rarely visit flowers that when I do the full analysis are not going to show up. Yeah, do you have a question?
So could you talk more about your, some categories, things like nestedness?
I don't know as much about that. So that's Casey's area. That's why I kind of glommed over that. I mean, I can, but a lot of it, there's a lot of questions. Basically, we found out from all that is bugs are such a small subset that I don't think it gives the full picture. That essentially was our result of looking at that connectedness. I don't know how much it is because just because a bug visits it, there may be, 7 species of bee that visit that same flower. That bug represents a small portion of the pollinators of that flower. That's essentially what we got out of that. So a lot of those terms and everything didn't really tell us a lot in the big picture. They told us that our picture wasn't big enough. Okay, so that's essentially my version of it as a field biologist, so.
And Angela, that's what I was sort of getting at.
Right.
There's a lot of words, but like, this is
Yeah.
the picture, so maybe, so I'm teaching a conservation biology class, and I think near the end, one of your adjectives is like fracturedness or
Right,
crumbliness.
yeah.
Or,
So if you remove a species, does it collapse?
and so is that a function of how many, that yours was like, not really, right?
Right. For bugs, no. I think it'd be different for bees because bees are so absolutely critical to them. So I think if we did the analysis for even just the bees, now, ideally, you'd want to be able to identify every one of those bees to species. And that's going to be another issue with my data set. The fact that I can't, that I don't have the time to be able to do that.
And so when you say we could remove a bunch of bugs and it would not affect the plants, is that because there are a lot that the bugs are on, the plants are visited by many bugs, and also because bugs are a small fraction of the pollinate, both of those things. Okay.
What I don't know is because that degree of specialization, it's like biennial gara almost always, every time I see biennial gara, I see stilt bugs on it. Do they have some other type of association? Are they basically secreting, something that helps the plant produce seed? I don't know. I mean, there's a lot of different factors that I have a strong feeling that there's probably some connection and some benefit that the plant may get as well as the insects. So there's a lot of mutualism that could be going on, especially for those specialists, and we don't know what that is. So for those, I don't think it's pollination, but I don't know what it is. And I'll probably, that's, who knows? Hopefully somebody studies that someday. I'd really like to know, especially in milkweed bugs, if there's something in particular, because they're so strongly associated with milkweeds. And everybody's planting milkweeds now for the help save the monarchs that, obviously those bugs are going to increase. So there's not going to be a concern for those bugs because they're going to do just fine. So they probably won't study those bugs. So any other questions? In the back.
So do you know, or could you talk a little bit about, say, if students were interested in getting to this kind of thing, whether like citizen science, right, there are observations through iNaturalist or someone else, that students participate, right? Not working, say, directly with the agency.
Yeah, yeah.
Collect data, right, and then at some point maybe be actually part of.
Sure. There's wonderful projects going out there, and they can find them on iNaturalist sometimes. We hold bioblitzes sometimes. I don't know if you've ever heard of a bioblitz. It's basically a lot of time. It can be a shorter one, but originally it's 24 hours of a particular defined area, like a site or nature preserve, all you can find. And we did one this last year, just here nearby on Wigwam Hollow Road. We only did it during the daylight hours. I did night lighting, that was it. We didn't do it till 24 hours because it's a lot of work. But yeah, you get a lot of different disciplines out there. Obviously, I'd love to do the insects because there's more. You never run out of insects in 24 hours. It's not possible. I don't care how small your area is. There's probably still gonna be more insects out there. So that's fun. I absolutely love to do that. I have tried to tailor this so that anybody can do it the way I do it. Will they find the same results as me? I've been doing it so long, probably not, but the methods are simple. They're very simple. I got them defined. I've got them out there. I have no problem sharing all this. And if somebody contacts me, I'll give them, my formal data sheets that I submit and things like that, and show them how I keep track of my data. But there's a lot of different projects out there. I know there's going to be a lot more bee research, but the more photographs that people can take, iNaturalist is great because it requires you to pinpoint where you saw that. Now, if it's extremely rare, listed species, they will blur, obscure where you found a rusty patch bumblebee. But for the most part, these that we don't know enough about, they don't know if they're critically rare yet. So you can actually pinpoint where you saw it. And then I can go out and see what you saw and other people can. And it's that community sharing is really important because when they all sit down, actually a lot of researchers are starting to use data from iNaturalist now because there's not enough of us out there looking for things. I still get a little bit of pushback. There are people in academia that think that photography is not a legitimate means of documentation, but it is what it is. That is what we have right now, and we're going to have to embrace it because we're not going to hire those taxonomists, especially if we're going the genetic route. So to me, that's the future. Photography is the future. We've got to embrace it without getting away with taxonomy. So any other questions?
So you're talking about looking at managed plots like, I don't know, a burn plot or something. Do you have an idea of what you might expect to see as in that kind of situation, like with succession or repopulation? Because it seems like bugs could come into an area really quickly.
Definitely, very quickly. One of the sites, if you've ever traveled to Cedar Glen or Kibbe Field Station on 136 here, middle of the way before you get to the the Lemoyne River there, up on the hillside on the right is Cecil White Prairie. We have been burning that, and that's one of my study sites. And so I have studied that one the 5 years, and I had about a almost 50% increase in diversity because of this increased management, because of these complete burns, because of brush clearing and opening up the prairie. So I'm seeing amazing results. The one thing that really surprised me is I wanted to know after the 5 years, am I finding the same species? And I'm not. So that's curious. Well, I've got this bigger list of diversity, and I'm seeing probably anywhere between 30 and 40% of the species I saw the first time I see again 5 years later. But I expected it to be higher. So that leads me to believe that I'm only getting a fraction of what's out there. Because I'm only there for an hour, 4 times a year. I'm missing a heck of a lot of stuff for all sorts of different reasons, environmental factors and whatnot. It may be cloudy that day that I visit. It may be for whatever reason that bug is not timed to come out at the time I'm there. So that's really curious to me. So there's a lot of things I can do with my data. So I just keep hoarding lots of data. We'll see where it works out. So. Anything else? Hi.
Agreeing with the prime element, these points sound very resilient, unfortunately, most of them are either not department, so they have options.
Yep.
Geographically, how many observations, whether they pollinate less or more depending on the environmental factors of a particular year, on wet or warm years, they shift their dependence on these plants.
I don't know that. I mean, I can speculate, but I really don't know. I mean, I'm only looking at like I said, I've just really gotten a lot better on doing the bugs. I just started looking at the bugs of the last 5 years. I think 15 years of data might tell me more about that. I know there are some bugs, not the ones I'm finding. I know there is a certain species of stink bug that was seen down in the Shawnee Forest back in the 80s, only on hill prairie remnants that they've not seen since. So there are bugs that we're disappearing that we just don't see. So it's not to say that there are not any bugs that are critically imperiled. That one may be gone. It may be extirpated. We may never ever see that one again, at least in the state of Illinois. So I'm sure there are some. Again, some of the problem is some of the plant bugs like Plagionathus, I can't identify the species. There's only a couple of them that are identifiable species. So there might be like 50 of them out there. And if I can't tell which species is doing what, I don't know. So there's always that factor. So there are definitely limitations in my photographs. So we will see what happens. Anything else?
Thank you so much.
All right.
Try to grab some more pizza on your way out, so you don't have to take it.
So we got a roll, so-
Yeah, I was, and then,
Okay.